All right. Just once again, this slide, your job was to come up with a early explanation or answer to this original scientific problem. So the scientific problem was, how do membranes play a role in removing solutes? And down in this little section, it's easy to miss, that's why I'm talking about it, is creating a hypothesis. And your hypothesis is really just an early answer or an effort to answer the mystery. So the mystery I'm asking you to answer is, how do membranes play a role in removing solutes? So your answer would be telling me confidently, remember it's a statement, it's a claim said confidently, you telling me what you think the answer is to this problem. So you would just say, my hypothesis is blank. Or what do you think is the role membranes play in removing solutes? Do they or don't they? You just keep it simple. Yes, they do. No, they don't. Membranes do not play a role. Membranes do play a role. You can keep it simple. And then remember, right down here, this lower part is at minimum. It says 4-sentence minimum. So if you write more, you get a better grade. But the idea is you write as much as you can, trying to decide. And in your writing, you're just trying to remind somebody, what my hypothesis was this, and that was the answer to the original problem. You can say the original problem, which is here, and you could say, the evidence suggests my hypothesis is correct because, or the evidence says that my hypothesis is not correct. So you can have an explanation here that's not right, and you'll still get an A plus grade. All you have to do is down here is say why the hypothesis was wrong. So no one cares in science if your hypothesis is right or not. It's up to you to say down here, using evidence, why it's wrong or right. And that's what I care about. And in the class, you're getting an A plus grade anytime you can decide if your hypothesis is right or wrong using evidence. So the real winning is being able to show why your hypothesis is right or wrong, not whether or not it was right or wrong. No one cares if you guess right. No, only the teacher cares about is if you can give evidence to support one way or the other. All right, good luck. I think I've talked about that enough. All right, let's move on. So this next slide is a repeat of the same thing. Just ignore it. And down here we have a situation. We did this last semester, but your responsibility is to relate this idea of diffusion. So you have a high blood alcohol content. You got pulled over by a cop. How would your body try to help you pass a sobriety test? So the question is the alcohol going to move from a high to low concentration? And where would it be doing that? What can the kidneys do, whose job it is to filter blood? So what does the alcohol want to do in your blood if your kidney's job is to filter the blood? Like, what would that look like? So you simply pick one of these choices here that shows which way alcohol moves. Does it move from high to low concentration or low to high? Pick the right one. All right, moving forward. We have this now. Osmosis is our last definition for the chapter, and it's just a specific way to look at water when it's diffusing, or when water moves from high to low concentration. Water, like everything else, moves from high to low concentration, and scientists have a special name for it because it plays a role in so many processes and cellular functions of the body, or of any organism to keep them alive. There's one other way you can think about this. You can always think about water moving from high to low concentration, just like you see in this picture. Lots of water molecules, and there's fewer over here. So the water moves from high to low concentration. It does. Another way you can look at this, if you get confused, is that water will also always move to an area that appears to have a higher solute concentration. This picture is a perfect example. This is pure water over here, and you have a lot of sucrose, or a type of sugar molecule, on the other side. The sugar molecule is trapped on this side, and there's lots of it. So it's a high concentration of sugar molecules. Water always moves, and you can see the arrow shows that. Water moving towards the solute. Water does this. In fact, a lot of meat back in the day, before we had refrigerators, was preserved by pulling all the water out of meat. That's how we got jerky or salted pork or cod. 300 years ago, the only way they could preserve foods is pulling all the water out of it. And they did that by coating meat in salt. And the water would run out of the meat tissue into the salt and pull the water from the meat. And it would preserve the meat. It wouldn't spoil once it became dry like jerky. Anyhow, you got 4 scenarios here, A, B, C, and D. And they all want you to consider this definition of osmosis, where water moves from high to low concentration. The examples C and D are specific to cells, where you have the inside of the cell and the outside of the cell. Here's some new terms, too. You will do an assignment out of the textbook that'll help you remember, but in general, hypotonic means a solution or fluid with very few solutes dissolved. So it's something that's a very low concentration. Here you can see the solute. There's only one solute in this surrounding solution. And then you have hyper, and that means more. So you have more solutes or higher concentration. So if you hear someone say hypotonic, it means the cell is surrounded in a fluid that has a very low concentration of solutes. If it's placed in a hypertonic solution, a cell has been surrounded in a high concentration or fluid surrounding the cell with lots of solutes. Anyhow, what's the solute in A? So example A, what's the solute? Which way will water flow in B? So you have water over here, water over here. Again, you may need to zoom in so you can better see the word. Oh, there you go. So you can tell the water is the blue circles and sugar are green circles. Anyway, moving forward, you just move and look at each situation. All right, we're almost done here. All right, this slide right here, I'm going to reduce the view of it a little. Okay, you have to collect some data, and I'm going to put up the simulation so you can watch it. The simulation on this video only runs for about, I'll play it for a minute. So you watch it for a minute, and then pause it. But before you, or I'll walk you through it, but you're collecting data. You want to know how much of a particular solute is on the left side of the cell, over here, how much solutes are in the inside, and the solutes on the outside. And the solutes are water and something else. We'll have to look at the simulation to figure out. But one of these is water, one's not. You want an initial number of solutes on left, inside, and right of the cell. And then after a minute, you're going to pause the video and count up how many are on the left, middle, and inside. Or sorry, left, inside, and right. Afterwards, you're going to discuss if osmosis occurred or not, based on our understanding of osmosis. We also have two different questions here I expect you to answer. But right now, I'm just going to help you collect data from this table. But remember, this slide, you're responsible for answering this question in the gray box, the question in the blue box, and collecting the data, and using the data, analyzing the data, making sense of it, interpreting it, or trying to understand what it means, and if it can tell you, can this data tell you whether or not osmosis occurred? And so you'd say, yes, osmosis occurred, or no, it didn't, and you'd provide your evidence. Your evidence would be your data. All right, so you just talk about, well, the results show that in the beginning it was this, finally it was that, so it shows that it did or did not occur. That's all you got to do. But use specific numbers here. Use the details from the data to support whether or not you thought it occurred. All right, let's not waste any more time. Let's go to the simulation. And I believe it's this next slide. Let's let it load. Again, if you're watching the video, there are links to all these things. So if you download the slides, you can highlight and go to this simulation, just like you see here. You can download. There's links on the slides, active links. Just copy and paste it into a browser like Google Chrome, and you can do the simulation on your own and use as much time as you need. So if this video didn't provide you enough time, that's what you need to do. All right, here, let's run it. Oh, okay, so the green circles are salts. So there's salt solutes on the left, middle, or so inside, and then the right side. You need to count the number of solutes on the left side, on the inside, and on the right. You also need to count the number of water molecules on the left side, in the middle, and on the right. Yeah, I know. It's going to take some time. So at this point, I would pause it. I'd pause the recording so that you can see this, but I'm hitting run right now. So if you haven't already counted the beginning of it, you need to rewind this video and get that number. All right, here we go. 3, 2, 1. All right, we're running this for 1 minute. Alright, so the question is, did osmosis occur? In other words, did water move towards the solutes, or did water move from a high concentration to a low? The only way you can know this, and let's go back to the slide to make sense of this all, is the final amount. How many of water are on the left side of the cell? How many of the solutes are on the left side of the cell? How many inside the cell, or how much water is on the inside of the cell? How many salts are on the inside of the cell? And how many salt, solutes, and water molecules are in the right side of the cell? All you then have to do is compare the initial amounts of water on the left, inside, and right side of the cell and decide, did water move towards a lower concentration? So where was most of the water in the beginning? And if most of the water was in a particular area, did it spread out and go to an area where lower concentration was? Or did the water go to where there was a higher concentration of solutes? Again, if you didn't collect the data as you needed to while the video was playing, rewind the video and get those numbers. Look at the amount on the left, middle, and right before I hit play, and then find out where I paused it after a minute and decide and get those numbers. How many ending or final amount numbers were there for the water molecules left, inside the cell, and right of the cell? And based on our definition of osmosis, or the diffusion of water moving from higher to lower concentration, and the idea of water moving towards solutes, did it occur? Yes or no? And again, the solutes would, in this example, would be the salt. So is water moving towards the solutes, or where the solutes are at highest concentration? Alright, you discuss these results here, you discuss this here, and you answer this question here. So there's 3 tasks to do. Record your data for beginning and ending. Use the results of the data to support whether or not you think osmosis occurred. Answer this question in the gray box, choose A, B, or C, and read this question in the blue box and respond to it as best you can. Alright, that's the end of the slide. Good luck. Make sure all these notes are in your book. And this is the end of Chapter 8 notes, so I won't be taking any more notes for Chapter 8. Thank you.